customer technology advisor at Resolve Biosciences. And we've seen at this conference already some great talks on spatial biology and how that technology field is boosting biomarker discovery and validation. And I'll take the next 25 minutes to also explain you what we are doing at Resolve Biosciences and how we differentiate ourselves as compared to some other technology providers. If there's anything that I want you to remember from my talk, is that we offer the highest data quality in the industry, and I'll explain to you how we achieve that. But first of all, molecular cartography is the technology we have commercialized at Resolve Biosciences, and it's an imaging-based spatial transcriptomics technology which is ideally suited for a hypothesis-driven research. So this is really downstream of your discovery phase, so really for biomarker validation in this case. And so on the right side, you can already see here actual data. So this is data from mouse small intestine, and each individual spot that you see here is an individual RNA molecule. So we do achieve that single molecule subcellular resolution in a spatial context. And so molecular cartography, as I mentioned, offers the highest data quality. So think about sensitivity, specificity, dynamic range, etc., combined with extreme flexibility when it comes to probe design or species compatibility. And also it's very easily accessible when you think about buying an instrument, or we have a very elaborate service program available as well. And so talking about data quality, so when we look at the technology space as such, we see a massive surge in these combinatorial FISH approaches, so imaging-based spatial transcriptomics approaches. When going from traditional FISH technologies with low multiplexing capacity, we now see technologies that can achieve very high multiplexing, so many gene targets per sample. But one of the things you always need to realize in any imaging-based spatial transcriptomics technology is that there's a direct trade-off between plexity, so the number of gene targets per sample, and data quality. So the higher you go in the number of gene targets per sample, so the more plexity when you're thinking about 500, 1,000, or several thousands of targets per sample, you inevitably decrease the sensitivity, specificity, and dynamic range of your entire assay. And one of the main culprits causing this is optical crowding. You're trying to discern so many individual fluorescent molecules in a very confined space that you're not able to optically resolve all of them. And this reduces the data quality across the entire assay for that particular cell or region of interest in your tissue. So at Resolve Biosciences, we really engineered our technology to achieve that highest data quality in spatial transcriptomics. And one of the ways we did that is we strictly adhere to traditional single molecule FISH chemistry, where we hybridize several dozens of probes to a single RNA target. And so you have that built-in probe redundancy, which on the one hand gives you that very high sensitivity, but also very high specificity. So with any mishybridization event or misbinding event, you won't pick that up in your optical setup because you need that accumulation of fluorescent signals in order for a spot to be detectable. And this is opposing some other technologies out there that use amplification-based FISH. So you're amplifying each individual signal, and of course, with any mishybridization or misbinding event, you will also pick that up in your imager, which increases the false positive rate in your data. And so next to the single molecule FISH approach, of course, traditionally you only have, let's say, a multiplexing capacity of 3, maybe 4 targets, and we engineered that technology to increase that to 100 targets per sample. Because we know with 100 targets per sample, we can really get to grips with that optical crowding and achieve very high data quality across many different species and tissue types. So it's really by design, not necessarily by limitation. So what we do is, after we prepare the samples, we design all these transcript-specific probes against all the 100 targets, and we hybridize it in a single step. And then we engineered our chemistry to allow for a specific fluorescent tagging of these transcript-specific probes, but we're also able to remove that fluorescence. And that allows us to go in with a cyclical imaging approach. And so that cyclical imaging approach allows you to go for higher multiplexing capacity. And the way it works is described in this cartoon. So Just an example for 3 gene targets, transcripts A, B, C. Of course, your list would be normally 100 targets long. And in the way we design our probes up front, we know that in the first round of imaging, for example, for transcript A, we know where it will light up in one of the 2 channels. So we use 2 channels and 8 rounds of imaging. So after capturing all those images in the first round of imaging, we decolorize the sample and we go to the next round of imaging, next round of colorization. And also the absence of signal is informative for our barcode that we are generating, but also we're not labeling all the molecules all at once in every round. And this is really allowing us to cope with that optical crowding as well. So again, removing the fluorescence, colorizing the sample again, going through all these different rounds of imaging, you see that after 8 rounds of imaging, you see that there's a unique barcode appearing. And the barcode is the sequence in which each transcript lights up in one of the 2 channels across those 8 rounds of imaging. And so after those 8 rounds, we can basically decode those barcodes. And all the spots we've been tracking in space in our image, we at that point can now replace with a transcript identifier. So the nice thing here is that our data is fully digitally quantifiable. So either a transcript is there or it isn't. So we do not use any fluorescent intensity measurements to quantify differences between cells or between regions. So full digital quantification. And so this brings me to another differentiator, how we achieve that very high data quality. And so this is an image taken from molecular cartography, and the big challenge in any imaging-based spatial transcriptomics technology is how to accurately define a spot. So the question you need to ask yourself here is, what is a spot and what is background? And this is where molecular cartography really differentiates itself amongst other technologies. So we're the only technology in the space that actually does a deep imaging. And I see that the left animation, right animation is not fully correct. So actually we have a full Z stack of 32 individual Z planes that we image. So we do a full deep scan of the tissue, and the result is that we actually see each fluorescent spot multiple times. Our spot calling algorithm is designed in such a way that it needs to see a spot as being faint, brighter, brightest, and faint again. And it takes all that information into account before it actually calls that spot. And so we are also not losing spots in between the different set planes in our imaging strategy. This is opposing all the other technologies out there that actually use sparse Z imaging. So you're, on the one hand, losing spots in between those set planes, so your sensitivity overall drops. You're not counting all the spots that are in your sample, but also you only have one chance of calling a spot or background. So that increases the overall noise and false positive rates in your data. On top of that, because we capture that full 3D information, we have a very accurate positioning of each individual transcript in full 3D in the data volume of your section as well. And so how does it look like under a microscope? So on the left side, you see molecular cartography. When you scan through the tissue in the Z-axis, you can actually clearly see almost with your naked eye what is a spot, what is background. On the right side, this is an example of merfish with sparse Z imaging, and it becomes much more difficult to know what is a spot, what is background. So generally, the result is that you will create much more noisy data. So both the single molecule fish and this imaging approach that we use results in the lowest false positive rate in the industry. So in any given experiment, our false positive rate is 0.05% compared to all counts. And so this is orders of magnitude lower than competing technologies. So the false positive rate for merfish, for example, is 5%, and for spatial molecular imager, which is developed by NanoString, it's 15%. And so this becomes very important when you think about biomarker signatures, where you're trying to detect very minute differences in gene expression. And so let's say you're detecting 1,000 signals in a given cell. 150 of those signals, when you would use SMI, they would be false positives, data that you cannot use. For merfish, this would be 50. And for molecular cartography, less than 1 or close to 0. So we're actually using this strategy, we're actually capturing the true biology in your tissue. So every count or every molecule that you're detecting is a true positive. So again, when you're looking at lowly expressed genes or trying to map out the different cell states in a tissue where minute changes in gene expression really matter, then every transcript that you're quantifying has a big impact, of course, and that's where that data quality comes in. And so how does it look like in an actual data set? So this is a cross-section through a mouse brain, and because our data is fully digitally quantifiable or fully digital, you can pick any gene that you want, give it any color that you want, and generate these nice looking rainbow figures. But of course, what you can also do is just within the same data set, select markers for inhibitory cells in this case. And in the same data set, you can zoom in all the way to the single cell level. And as you can see, just based on one marker, you can actually see these somatostatin positive interneurons in yellow, GAT1 positive interneurons in red quite easily. And within that same data set, you can dive all the way to the single molecule level in full 3D. And you can do actual transcript counting. So you can count each individual somatostatin RNA molecule in this cell in full 3D. So if you're interested in mapping differences in the distance towards the center of the nucleus, for example, to investigate RNA polarity, that's also possible with our technology. And I want to make you aware that we're able to detect molecules above the nucleus, below the nucleus, and everywhere around. So this is really our approach to 3D spatial biology, where you're capturing all the molecules in your tissue section. So really increasing that sensitivity and specificity. So this brings me to flexibility. So we offer 100 genes per sample, but you can fully customize your gene list. So you can really tailor your gene list to your research needs. And it's not only limited to, let's say, polyadenylated RNA, but you can also target long non-coding RNAs or fully custom sequences and much more. So we're the only company in the space that offers the highest species flexibility. So we have protocols available and generated data for more than 14 different species, over 50 plus sample types. So of course the typical mammalian sample types or species like of course human, rat, mouse. We go way beyond that. Model organisms, zebrafish, Drosophila, Xenopus. It's compatible with cell culture, organoids, and we are very active in the agrobiotech industry as well, with lots of protocols on plant samples. And so in terms of flexibility, when it comes to experimental design, we have a glass slide with 8 different positions. Each position is around a square centimeter in size, but it allows you to quite easily generate an experiment, for example, with 4 controls, 4 treated patient samples, for example. And this allows you to easily keep track of where you placed which sample and generate or design your experiment in such a way. And throughout the entire molecular cartography protocol or assay, you're capturing all the transcriptomic information, but the sample remains intact. So we're not using any proteinase K or clearing reagents during our protocol. So what it allows you to do is go in with the downstream immunohistochemistry or other types of downstream analysis. So we have developed a universal protocol that is compatible with your own antibodies. So you don't need to buy antibodies from us or conjugate your favorite antibody with an oligonucleotide or new fluorophore. So you can actually use the antibodies that you have validated and lying in your fridge. Other users, for example, have done DNA fish. We also have protocols for H&E and much more. So all the content in your tissue remains intact, which allows you to add extra data layers to your data set. So it's within the same tissue sample. And I'm very happy to announce as well that we are now fully offering full compatibility with FFPE samples as well. And this is a data set on human colon cancer, 23 genes, with a very low RIN score. But despite that low RNA quality, you can actually see that the data that we generate with only 23 genes is very dense. It's very reminiscent of what we would expect also with the fresh frozen tissue. And so we're developing these FFPE protocols, of course, focused on human, but also mouse and plant samples. And we offer that same data quality. And with the data quality, I mainly mean the false positive rate. So with matched fresh frozen and FFPE samples, we see again that extremely low false positive rate appearing. So we're also capturing that true biology in FFPE samples. And when comparing the number of counts in matched FFPE and fresh frozen samples, we see an extremely high correlation of 0.94 R square value. So of course, you always expect fresh frozen to be slightly more sensitive in a single molecule fish-based assay. This is also what we are seeing here. But a very high correlation is, of course, showing you that you can capture that high data quality as well for FFPE samples. And so how can you access molecular cartography? Well, there are many different ways of accessing it, and we support you in whatever way that you choose. So you can, of course, purchase our instrument, but you can also access molecular cartography through third-party service labs. And a lot of people access our technology through our very extensive white glove service program, which is called Meridian. So using Meridian, you can go from sample to data in a matter of weeks. So our typical turnover time is around 6 to 8 weeks from sending us the samples to getting the data back. So how it actually works is we have a very extensive online probe design tool where you can design your own experiment, you can insert all the gene targets that you want, of course under guidance and support from our experts. We send you glass slides, you prepare the tissues or the sections, and you send it back to us. And we do everything else. We perform the entire experiment, and we set up a dedicated meeting with you and provide you a full data report with QC metrics, and we also offer additional bioinformatics support to really get to your biological insights very fast. And so using or having this service model already in existence for a little more than a year, we've seen the fastest publication rate amongst all spatial imaging providers. And so this is really a testament that molecular cartography is rapidly being adopted by many researchers, by the entire research community. We now, at this point, have 18 high-impact publications across many, many different applications. So it's really nice to see that molecular cartography is fueling so much of different branches of life sciences. And so coming back to that data quality, flexibility, and accessibility, this is really fueling a lot of different applications. And again, so also, focusing on biomarker validation, that data quality, I think, is extremely important, where you're trying to quantify sometimes very minute differences between cell types or ingredients in a tissue, for example. And so with the rest of my time, I want to briefly go over one of the applications. So this is situated in liver. We have extensive experience with human and mouse liver in both healthy and diseased context. And I want to highlight a recent publication by Paul Monga. So he's the director of the Pittsburgh Liver Research Center. And so he's interested in liver zonation and regeneration. And he, in this Cell Reports Medicine paper, he extensively used molecular cartography to validate many of the hypotheses he had. So he used molecular cartography first to validate that you can indeed in mouse liver see these gradients going from the central vein to the portal vein based on unique markers. And he's mainly interested in Wnt signaling. And so, of course, he validated that he could see specifically for his research needs Wnt2 and Wnt9b expressed in the central vein zone. And it co-localized with PCAM1 as a marker for endothelial cells, also with other markers, but he was specifically interested in these two Wnt molecules or targets expressed in endothelial cells and what they achieve in liver zonation and repair. So he actually designed a conditional knockout for Wnt2 and Wnt9b, specifically in endothelial cells, and he used molecular cartography to actually validate, again, that there's a loss of zonation, specifically around the central vein. The portal vein regions seem to be relatively intact as you compare that to controls. The nice thing about the spatial data set is you can always retrieve single cell data from that. So this is also showing you what you can do with this type of spatial data. So he segmented each individual cell, extracted the transcriptomic profile for each individual cell, and generated these UMAP clusters. And again, you can see that specifically for the central zones, or the zone surrounding the central vein, you can actually see a loss of zonation. So these hepatocytes, these cells around the central vein, they don't express the proper signature anymore after Wnt signaling knockout. And so he went another step further in how to quantify the spatial data, and he used some landmark genes to identify the central vein and the portal vein, and then he drew several compartments going from the central vein to the portal vein to actually map differences in gene expression in a spatial gradient by counting individual spots and normalizing that to the surface of the zones. Actually, he divided that into 9 zones. And this allowed him to really characterize with very high accuracy different gene signatures underlying the loss of that zonation. So not only focused on the central zone or pericentral zone, he also did the same thing for the periportal zone and really mapped out the dynamic changes in gene expression when comparing the control and the double knockout condition. And this shows you a little bit what you can expect from this type of data. You can also, for example, include other genes related to specific pathways and really think about it in a spatial pathway type of analysis. And so lastly, in this study, he combined this type of analysis and molecular cartography in an animal model where he did an acute liver injury using a high dose of APAP treatment, and he also was able to rescue that loss of zonation. So after APAP treatment, there's a massive injury in the liver, and you lose that zonation again. And with an FL6 antibody, which is basically an antibody that stimulates Wnt signaling and beta-catenin signaling, he actually saw with molecular cartography, he could validate that there's a restoration of that zonation happening actually. And so this is a very nice demonstration of why and how you can use spatial biology to validate the involvement of Wnt signaling and also validate this as a target. So I want to end here. So if you're interested to learn more about molecular cartography, we're here also with a booth, so booth number 3, just outside here in the exhibitor hall. So we're happy to learn more about your interest in spatial biology, how we can apply our technology to your biomarker research needs. So thanks a lot.
Thank you very much.